Hello and welcome to your first lecture and video notes of the semester. Pretty straightforward, and you've probably heard a lot of this before, so it will serve as a review, but I want to make sure that we fill in any gaps, and I also want to make sure that we maybe turn up the sophistication of your understanding of a couple things that you may have learned at a more simplistic level earlier in your educational career. Now, before I get too far into this, I want to make sure you understand that this is where we are. We're on this page, the first thing that you're doing in your packet. Okay, you're going to follow along with that, and that's going to help you understand and retain the notes that we're doing here today. All right, so let's get started. Laboratory structure is what we're talking about in the wake of your first mini lab, which was for practice, the 8 solutions lab. Over the weekend, you have a second practice lab. This one, you're actually going to turn in a full lab to me, although it is a completion grade. And you're not going to be marked down if you make any mistakes. All right, laboratory structure. All right, so in broad terms, I want to make sure that you understand a little bit more about a scientific mindset than maybe you may have learned from more simplistic instruction earlier in your lives. And if you have more sophisticated instruction at lower levels, fantastic. I'm very happy for you, and it'll make things a little bit easier as you go through this course. That's a good thing. We've seen this last 6 months or so that scientific processes are not always neat and clean. Scientific processes are often very messy. In fact, nearly always. It's just that the public doesn't always see it as it's ongoing the way we have with the science surrounding COVID-19 over the last 6 months. There were mistakes made by scientists. There were choices made before there was enough information by some politicians and other policymakers. And the understanding of the virus has evolved over time and is still evolving. And so while we've gotten a pretty good handle on the actual keeping people alive part, and yet there are still people who are dying, and I don't want to diminish that. What we're learning now is that even when we can keep people alive, which we continue to get better at, there are long-term consequences from this virus that nobody anticipated and that are just really now beginning to come to light and being studied. And so that's part of the reason why COVID-19 is so important and why things are different here than it would be if this was a new flu virus or something like that. The flu doesn't cause scarring of the lungs, blood clotting issues, cardiac issues, kidney issues, and neurological issues the way that COVID-19 can. And that's why we're taking these extra precautions. But yet we didn't really realize that until the last couple of months. So make sure that you understand that this is what science is like. It doesn't mean that the scientists are full of crap and can't be believed. It means that they're working on it. Scientific understanding doesn't come like that. You don't just look at a pile of data and all of a sudden, hey, I know how that works. It takes time. It takes time for us to really get a handle on things. And every major scientific development throughout history has gone through the same kind of messiness that we saw. So I hope you understand that. And that's why I want you to know that scientific processes don't often go the way that the scientific method is presented, particularly in elementary and middle school. Like you do these 5 steps and then you have results and your experiment is done. And it's not like that. A lot of times you get to step 2 and realize something's wrong and you have to go back and repeat and start over, and that happens a lot. And you repeat and start over. And then you realize that you only have maybe a piece of the information. You need to design a broader experiment or a different experiment, and you repeat and you start over. And lots of different people doing this can seem very messy, especially at the beginning of something, before it begins to converge towards a deeper understanding and a clearer understanding. And we continue to make progress on that. So what's important to understand is that we often repeat things, sometimes because that's what's necessary to understand, and sometimes because that's what's necessary to verify information. So even when I do finally get to a nice, clean experiment with really clear data, someone else is going to go and try to duplicate that experiment, and they're going to try to verify the results. If they can't get the same results using the same procedure, then my information is suspect. And so there's a lot of repetition or iteration that goes on in science. Despite the messiness, though, scientific processes need to be systematic, which means that you have to follow a plan. And you might double back and revise your plan maybe several times in a large-scale professional experiment. But There is a plan by which we proceed. There's an order in which things need to happen. And the messiness, the looping back, the repeating, and the trying again has to do with making sure we get that step correct before we move on to the next one and continue. And a lot of it is trial and error. We know a lot of things about the world, but there's a lot of things we still don't know. And we have to try based on what we've already learned when we go forward. Still, you've probably seen a lot of these things: hypothesis, experiment, analyze data, draw conclusions. Notice that the hypothesis is not at the beginning, and that'll be true in my labs, in the actual structure of my labs as well, and that's very important. A lot of times when students come to my class, what they think is a hypothesis is actually just an initial question. An initial question is something that you ask or are asked at the beginning of a lab before you know anything. Maybe we talk about the last topic. I do that a lot with labs. I'll ask an initial question that's related to what we just did, just to review it and refresh it in your memory. A hypothesis is not a question that you can't answer, and it's certainly not a question that you try to answer before you know anything. A hypothesis is informed. And in order to be informed, you have to start learning about something first. And so hypothesis comes after conducting research in the professional world. We don't do research in the chemistry lab, although if you go on to take one of our higher level sciences, BRED, biological research and experimental design, then the whole process is modeled in there. And trust me when I say their experiments are messy, just like we talked about. So research comes first, then you formulate a question, and then initial investigation, which could be more research, but often is trying out the experiment or little bits of an experiment. How will this work? How can I gather the data? What might work for me to be able to learn something about this? So there's a little bit of investigation. You'll see this with our labs in units 2 and 3, where you actually experiment first with one thing, form a hypothesis, and then go on to the final part of the experiment. A hypothesis should be a declarative statement. You need to make it specific. It should say, this is what I think is going to happen. It doesn't say, well, maybe. You don't hedge your bets, and you certainly don't make a hypothesis after you've already found out what happens. That's cheating. You need to predict what's going to happen based on what you already know. If you're not thinking about what's already happened in the experiment or what's already happened in the unit for the class that the experiment is in, then you're probably not going to have a very good hypothesis. So make a statement, make it bold, and say, this is what I think is going to happen. This is what is going to happen from this experiment. If you're wrong, it's fine. You don't lose points for a wrong hypothesis. You lose points for a weak hypothesis. So make sure that you understand that. And I will provide guidance as we go forward. Hypothesis should be specific, so don't give me just general terms. It should be targeted directly to what we're trying to find out. Sometimes people make a hypothesis that's tangentially related rather than directly related to what we're doing. And it needs to be testable. In other words, does the data that we're going to gather actually tell you whether your hypothesis is right or wrong? So that's important. Now, related to the concept of hypothesis, I found that not only do a lot of people not fully understand what a hypothesis is, but a lot of people out in the public domain, including many adults, don't really understand what the word theory means. When people say things like, I've got a theory, or that's just a theory, now, what they mean in those circumstances is idea. I have an idea. Well, that's just your idea. A theory is not just anything, and there's no such thing as just a theory. The theory is actually the highest standard that scientists use. So let me give you an example with something you're familiar with. Gravity. So there's a law of universal gravitation. Isaac Newton, among others, described the law of universal gravitation mathematically way back in the 1500s. So for nearly 500 years, we have known what gravity does. That's all a law does. It describes what happens. It is the cohesion of a number of observations that brings about a consistent understanding of what's going on. But a theory is much more than that. And so there is also a theory of universal gravitation. And that theory of universal gravitation is still very much a dynamic, living concept that is being worked on by physicists all over the world as we speak. It is one of the frontline current topics of modern physics. The understanding of why gravity works. To be able to explain why something happens, how it happens, and how it's connected to other things in the universe is a much bigger deal than to be able to explain what happens or to demonstrate what happens. So theory is at the top of the charts. The theory has the broadest and the deepest amount of evidence for it. Something doesn't get to be a theory unless there's already enormous, significant evidence that explains what's going on. That doesn't mean that we don't change or adjust our thinking based on new evidence. What it does mean is that we don't develop a theory with a ton of evidence and then all of a sudden there's this huge tidal shift in our understanding. We make small corrections based on what we know. And so a theory is very, very strong. There's no such thing as a weak theory in science. And I want you all to understand that. It's important to use words correctly, and I like to emphasize that in my class. That brings me to an important point here that I want to talk about. The difference between a scientific mindset and an ideological mindset. And I don't tell you this just for science. I don't want you to just turn on the scientific mindset when you come into science class or when you're working on science outside of class. I want you to have a scientific mindset everywhere in your life because I think a scientific mindset leads to better understanding. So let me explain what I mean. To have a scientific mindset simply means to look at the evidence first and then decide what you believe about it. Look at the evidence and then interpret it. Look at the evidence and then make your statements about it. That's a scientific mindset, very simply stated. An ideological mindset, on the other hand, decides what to believe first and then looks for evidence that supports the belief that has already been chosen. And it is an ideological mindset that often leads people to dismiss and discard contradictory evidence, things that challenge the beliefs that they've already decided to hold. I don't think this relates only to science. As a matter of fact, I think it relates to everything in your life, up to and including your religious beliefs if you have them. Let me give you an example. Should you decide what to believe and then go look in the religious texts to find the evidence that supports your belief? Or should you read the religious text first, talk to people who have studied it, gather information, and then decide what to believe? Which one leads to deeper understanding? I think it's important to have a scientific mindset, to not have an ideological mindset. We'll touch on that one more time before we're done here today. All right, so the last thing I want to talk to you about in terms of lab structure is the actual experiment. There's a couple things we'll touch on very briefly after the experiment, but we'll talk mostly about the experiment here. I want to talk about how you set up an experiment properly. We're not going to go into tremendous detail, and you will only once be asked to design your own experiment in class. That'll be near the end of the year, and that's just to demonstrate that you've learned these processes through what we're talking about here today and through your experiences with labs. Of course, if you go on to higher level science courses, you will often be asked to design your own experiment, and so that level of thinking escalates as we go forward through our educational career. First of all, hopefully you've already heard of independent variables and dependent variables, right? So an independent variable is the one that I choose when I'm designing the experiment. I set the values. I know exactly what they're going to be when I go into the experiment. They're already known. The dependent variable, on the other hand, is the one that I'm trying to find out. That's what I'm going to be discovering as I go through the experiment. I don't know what those values are. If I've developed a good hypothesis, I should have a good idea of what my dependent variable is going to do. But you never know for sure. And science can give unusual results sometimes. Even when we've got everything lined up perfectly, we very rarely get perfect results out of a scientific experiment. And so the dependent variable is always a little bit of a mystery. The dependent variable depends on how I choose the values for the independent variable. So for example, if I want to measure the temperature outside every hour over the course of a day, right, then I would choose the time on that. I've chosen hours. That means hours is my independent variable. 1 hour, 2 hours, 3 hours, and I'll record the temperatures. What I don't know is what the temperatures are going to be. Now, I may have a good idea of what they're going to be, But as you all know, sometimes the temperatures don't match the forecasts that we have, even within an hourly basis, let alone a day in advance or more. And so we discover what the temperatures are going to be at each hour, which I chose. Right? And so this is connected to graphing, which we're going to do a little bit next week. The independent variable goes on the x-axis, the horizontal axis, and the dependent variable goes on the y-axis, the vertical axis. And we say y versus x when we're talking about the name of a graph or when we're describing the relationship between these. So in my case, it would be a temperature versus time graph, or in a more sophisticated way of saying it, temperature as a function of time. So the dependent variable is a function of the independent variable, at least as long as they're connected the way we think they are. Now, the rest of this that we're talking about on this slide has to do with controlling all the other variables. Now, control has a double meaning in science. There is a control sample in many experiments. What that means is a fixed standard that we will compare to. Right? So, for example, I might have a set of temperature data from yesterday, and I would use that to compare today's temperature data. That's how forecasts are made, after all, historical data being applied to current situations. So a control can be a standard for comparison, but controls are also the ways in which we eliminate confounding variables that might give us the wrong answers, the wrong results, or the wrong interpretation of what we're doing. And so we have to isolate our independent variable and our dependent variable to the best of our ability and make sure nothing else is changing. And in most cases, that is really impossible without sophisticated, expensive equipment. But we do the best that we can. We certainly don't want to have 4 or 5 things changing during the course of a lab. Then how do we know which one caused which effect, or how much each of those changes is responsible for the outcome that we have? And so that's what experimental controls and the idea of experimental design are about. I'm not going to spend any time on analysis. There's just 3 common things that we do with that. That's after the data has been collected: calculations, arranging charts and tables, and graphing. You're going to see those on a regular basis this year. There are higher level statistical methods that can be used for more advanced sciences. And finally, at the end of the lab, we draw conclusions. Drawing conclusions involves 2 things that almost always happen. One for sure, and a second one that nearly always is in the conclusion section. So when you write your first conclusion statement or conclusion paragraph next week, these are the things that are going to be in there, and always in there in my labs. But first of all, you evaluate your hypothesis. Was your hypothesis supported or refuted? And it may not always be clear. This isn't always a either or binary situation. Sometimes your hypothesis might be partially supported. It's okay to explain how some parts of what I said worked, but other parts weren't what I predicted. That's all right. In fact, that often leads to a more detailed breakdown of what went on during the lab. And more detail is good when we're talking about these things. So you talk about your hypothesis and evaluate it, and it's okay if your hypothesis was wrong. You do not lose points for wrong hypotheses. You lose points for weak hypotheses. So make sure that you understand that going in there. I don't care what your hypothesis is, as long as it meets the criteria for a hypothesis. And then finally, in most labs, we will look for sources of error. During your measurement lab, which you're going to look at over the weekend, and we're going to discuss on Tuesday, I pointed out that I can spot as many as 3 really easy common errors that could happen during that lab, that may have happened during that lab. And we're going to discuss those in class on Tuesday, so make sure you're paying attention for those potential errors. Sometimes you also suggest directions for future research or how to fix the experiment. So we might, in a conclusion statement, I might ask you, how would you change what we did to make it work better? Or what would your next experiment be? And I do reserve the right to ask extra conclusion questions sometimes if they're important. Finally, a scientific report is not complete without a purpose statement or an abstract or an executive summary. Now this year we're not going to be putting much emphasis on this. That's the one concession that I'm making to COVID-19 when it comes to our lab reports this year. So the idea of a purpose statement is to summarize what you were supposed to learn in the lab, along with what you actually did learn about that. In my class, the purpose of a purpose experiment or a purpose statement is to connect to the material in the unit. So a lab is not separate from the material that we're studying. A lab is directly connected to the material that we're studying in that unit. And the purpose statement is for you to make that connection clearly, explicitly, and with detail. All right, and so this is my final slide here, and I just want to revisit one more time the idea that scientific processes are not always a nice, smooth, straight line as they're sometimes presented. Sometimes things go wrong and we have to go back and change our approach. Our systematic approach needs to be different. So for example, when you first design an experiment, looking at the light blue in this chart, the first thing you want to ask is your procedure working? Which is not asking the question, did I get the right results? What I'm asking there is, did my equipment function properly? Did I actually gather the data correctly? Or were there errors in the way the data was collected? Is the data faulty? And so if that's the case, you have to go back and fix your procedure first. So you loop back and you try a different procedure. And oftentimes that happens several times before people develop a correct procedure, especially when they're experimenting on something that is new enough that it requires its own novel procedure. And then finally, after we do get good data, good meaning not necessarily right, but it means that it comes from a good procedure, it comes from a good process. When you get good data, then it has to be analyzed. And if that analysis means that your hypothesis was wrong, well, then that's when we talk about the scientific mindset versus the ideological mindset again. Right? So if I get data from an experiment and it says that my hypothesis was wrong, I have two choices. I can either discard that data, assuming that my hypothesis is correct, and then go look for more data to support it. And sometimes, unfortunately, this even goes as far as manufacturing data to support it. And those types of rare studies, it doesn't happen very often, but those types of rare scientific studies where the data is manufactured to support an ideological belief that was already formed are very damaging. They damage by leading to false information, and they also damage trust in scientific institutions. And it's important to be aware of those things. On the other hand, if I find out that my hypothesis is wrong, I can choose to go back and revise my hypothesis. I can go back and change it. I can change what I believe based on the evidence that I've learned and keep working until I have a good understanding, until my belief coincides with the evidence. And so I submit to you that that's the way you should be approaching life as well. And I hope you will work towards that. Systematic thinking is not always easy for teenagers in general, or for people overall. Some people think naturally, more systematically than others. You're going to learn that, though, and you can develop the skill of learning, of thinking through things systematically, of approaching and solving problems systematically. You can train yourself to do that. And it's one more way of thinking, one more way of approaching the world around us that can help you gain understanding. And so this is much more than just a scientific process. This is another way of understanding life. And I hope you'll take it as such. Thanks for listening to me today. If you're curious about more details, if you go to the actual notes file, you can find a link here to more detailed description of the true scientific method, not the simplified one. And I'll also tell you, earlier in this set of slides, there's a little Easter egg. There's a link hidden in there. See if you can find it. Let me know when you come to class next time. Thanks for listening.